course, one of the major secrets to being a successful college student is managing your time effectively. Here's an old Calvin and Hobbes cartoon. Is it, Hobbes says, "Here's a nice leaf. Do you want it for your collection?" No. Why not? It's a beautiful afternoon. I'm not going to waste it doing ridiculous busy work for school. But this would be one less leaf you'd need to collect later. Yeah, but it's one more leaf than I need right now. Somehow it's always right now until it's later. Whatever that means. So even when you have an opportunity to do the things you need to do, it's real easy to, I don't need to do it right now. I'll do it later. I can do part of it now. It's just part of life. So we're following up on developments in genetics. I thought I'd hit the end of the history, and then I realized I hadn't. There's a couple of things still to put on there. This one is incredibly important. PCR. When people started to come up with ways to match DNA by looking at select pieces that, when taken in combination, were fairly unique to individuals. I'll talk about this in a couple of minutes. The thing is that you needed an awful lot of DNA to be able to analyze it. And so it wasn't really useful for things like crime investigation, like forensics. There was no way to take a small amount of DNA and do anything useful with it. It was just too limited. Well, someone who actually just died recently developed a method of taking DNA and what's called amplifying it. Basically, taking a small sample and making lots and lots and lots and lots and lots and lots of copies of not the whole genome, but pieces, important pieces of the DNA that then give you lots to analyze and compare. Well, how did they do this? They developed a polymerase chain reaction, also known as PCR, which is also part of the major COVID test, the one they stick way up your nose, is a PCR reaction where they actually amplify RNA. But anyway, and we'll talk about the details of this way at the end of the second semester. Here I'm just going to give you the rough outline. He found a hot springs bacterium and isolated the enzymes that it used to make DNA. Now, DNA doesn't like extreme heat. Enzymes don't tend to like extreme heat. They lose their shape. The heat shakes them, they fall apart. But of course, if you've evolved in conditions of hot springs, you either are there alive and doing stuff, or there was nothing in the hot spring at all. And there were living things there. Thermophilus aquatus, I think. TAQ. TAQ polymerase is what they use for this. And it is an enzyme that can make copies of DNA at high temperatures. So here's what you do. And as I said, you don't need to know the steps, but I can't help myself. I'm going to tell you the steps. And then late in the second semester, you need to know the steps. You take a small sample. Figure out what you're looking for. You can make copies of the whole genome, but it's not easy to do, and you're going to get contamination, a lot of stuff. And that's something also true. Your samples often will have contaminants in it. And so it can be very tricky to actually do DNA analysis and get it to work right. But you put your sample in, you heat it up, the two strands of DNA will come apart, you heat it down, not cool enough that they'll reassociate, but cool enough that you hit the temperature that this enzyme will work at. You also hook onto the DNA a little piece of one side that will attach to, when you open it up, it'll attach to a particular spot, and it'll attach to another spot. What they call primers. That will allow you to make copies of not the whole DNA that you put in, but just these pieces that you're interested in. So you heat it up, drop it, get it, put the primers on, heat it up a little bit, expose it to this polymerase, and what the polymerase will do is, starting from one primer and working to the other primer, it'll make a copy of what was in between. So it'll make a copy of stretches of DNA, and then it'll do that to all the DNA you put in there, and then you heat it up, the DNA comes apart. You put it back down, you put the primers back on, you heat it back up, and now your extra pieces that you just made, you make copies. And you just keep doing that. Actually put it in a machine, the machine keeps doing it. Just raising and lowering the temperature. You've mixed it in with a bunch of the right reagents, the enzymes and stuff. And it'll take a small sample of DNA and make a very large sample of select pieces of DNA and allow you to analyze, like, you know, cells off of a comb or a little drop of blood that was left at a crime scene. It's gotten very, very, very useful. Extracting DNA from ancient teeth, although I gotta say I'm a little skeptical of some of the stuff they're doing with really, really ancient DNA. Is that sometimes the problem is that this is so automated and so mechanical that we're trusting the observations of some devices that I'm not sure are quite as precise as everybody would like them to be. But they write papers on it like they were really, really precise. Now, I've been trained in PCR, but I don't really, I haven't been trained in the really, really advanced stuff that they're doing now. So I'm, you know, you probably should not listen to me about whether to trust this stuff or not. I'm just telling you because, you know, I'm talking into the screen. I don't necessarily trust some of the more extreme versions of tiny, tiny pieces of DNA, or tiny old pieces of DNA. And this brings us to what I was. And again, this is something we'll do the second semester, but you take your mixture of DNA, it's different pieces. You've put primers on, different pieces and made a bunch of copies, but now you want to separate them out. And you put them into a material that they will migrate through, and then you put electricity into the material, and it migrates, different pieces migrate to different, toward the other charge. Different amounts. You do it for X amount of time, you stop it, you stain the medium to make the DNA show up, and you will get band patterns. You know, you'll get band patterns that I think they use 13 of them in doing forensics DNA analysis. And the combination of bands is supposed to be pretty reliable for matching DNA from one source to DNA from another source. The numbers that they throw out, you know, what are the odds of this blood coming from someone other than the defendant, Your Honor, and the guy on the stand will go, oh, it's like 1 in 240 million. Nobody knows that. Is that it's all guesswork because they haven't done enough of the testing to know really how rare or common it is. And they're also much more inclined to ignore some of the vagaries you get when you actually run this in the real world, to go, oh yeah, those definitely match. And sometimes it works like fingerprints, where you'll actually get, once you put stuff into the DNA database, you may get like 3 matches, but only one of them makes sense. Only one of them is local. And so you go, well, forget the other 2 matches. They do the same thing with fingerprints to some extent, where it's not always a 1-to-1 match. But this is a really big deal for all sorts of different types of testing where they're using bands. And then the very last thing on our list, we've talked about this before, but it's become a really major part of genetic studies in the last 15 years. It's been around for a really long time. But the idea of gene activation and deactivation. How much is a gene activated? How much is it deactivated? Does it stop entirely or does it slow down a little bit? This is basically the study of genetic control, where right now you can't have every gene activated in your cells. Your cells would be producing all sorts of proteins not supposed to be working at this moment. But how is all of this system at play? And that's become a really, really, really major part of a lot of study right now. I mean, it's a really big deal for cancer. What happens in cancer is you may get mutations, but basically a lot of times what the mutations do is they either shut off a gene that's supposed to be controlling the cell, or they turn on a gene that wasn't supposed to be turned on in the first place. The cell keeps dividing. But it's an activation issue. Even if there's a change in the DNA and it's a mutational issue, you're often talking about epigenetics as well. Now what used to be called epigenetics is now called mimetics. I'd mentioned that before. Is that mimetics specifically looks at things that are genetic technically because they're inheritable, but they're not gene driven, they're not DNA codes. Like English is a mimetic inheritable feature that people teach you English and you teach it to other people, and you can pass it on and you can change it on, pass it on in an altered form by writing stuff down in a new way. But culture is mimetic. Lots and lots and lots of different things are inheritable, not just person to person within a species, but a bear passes particular food territory onto its offspring. That's inherited. It may give them an advantage in the world that they might be able to pass on to their offspring, but it is not in their DNA. And so epigenetics and mimetics are now different things, where what we now call mimetics used to be called epigenetics. So if you look at an old source, that's going to be a confusing term. Okay, right here is the cutoff for the first exam. Is that we're not done with the genetics chapter, but I'm going to pick up the rest of the genetics chapter after the exam and add in some of the new developments. But this is basically the history. And the history I want on the exam, the later stuff I want on the second exam. So it's going to be posted that way, but don't be surprised when after the exam, what comes up for lecture is still chapter 5.